The overall goal of this protocol is to observe the contribution of proton transport to the rate of bacterial extracellular electron transport by the detection of the deuterium kinetic isotope effect.
Our method reveals the impact of proton transport on the kinetics of extracellular electron transport, or EET, from bacteria to an electrode via outer membrane sheet-type cytochromes. Our assay reflects the native proton management in EET because we use living bacteria rather than purified enzyme. However, our challenge was to remove the noises from other cellular processes.
The Deuterium Kinetic Isotope effect, termed as KIE, is one of the best techniques to examine the contribution of proton transport on the associated electron transport process.
To grow Shewanella MR1 cells, transfer one colony of the cells grown on an agar plate into 15 milliliters of LB medium. Grow the cells at 30 degrees Celsius for 24 hours in aerobic conditions with shaking at 160 RPM. Centrifuge the cell suspension at 6,000 times gravity for 10 minutes. Then, resuspend the resultant cell pellet in 15 milliliters of defined medium, supplemented with 10 millimolar lactate as the source of carbon and 0.5 grams per liter yeast extract as trace elements for the cells. After further cultivating the cell suspension aerobically at 30 degrees Celsius for 12 hours with shaking at 160